It is an honor to introduce the latest research results published in iMetaOmics. Through combined transcriptomic and microbiomic analysis, the study revealed that the key influence of microbes on immune modulation and disease progression in colorectal cancer. First, let's understand the core findings of this study through the graphical abstract. The study identified two distinct colorectal cancer subtypes, CA1 and CA2. The CA1 subtype shows an immune-active but exhausted state. Although it has high immune cell infiltration, the high expression of immune points and enrichment of specific pathobionts, such as P. gingivalis, lead to a poorer prognosis. In contrast, the CA2 subtype, despite having lower immune infiltration, is dominated by health-associated microbial communities, and patients show a better survival prognosis. This suggests that microbes play a key role in reshaping the tumor microenvironment. To investigate the host microbiome interaction, we collected 31 pairs of tumor and adjacent normal tissues. Notably, we used 2B-RADM technology, a high-precision sequencing method that effectively addresses the challenge of detecting low biomass samples, such as in situ tumor microbiome. By integrating host gene expression and microbial functional genes, we built a co-expression network and identified a core immune-related module, the M95 module. Based on the expression profile of this module, we classified tumor samples into CA1 and CA2 categories. Clinical correlation analysis showed that the overall survival rate of patients with the CA1 subtype was significantly lower than that of the CA2 subtype. By further analyzing the immune microenvironment, we found an intriguing phenomenon. Although the CA1 subtype exhibits high levels of CD8 T cell infiltration, appearing as a hot tumor, it simultaneously highly expresses immune checkpoint molecules such as PD-1 and CTLA-4. This indicates that T-cell CA1 subtype are functionally exhausted, which explains why high immune infiltration is accompanied by a poorer prognosis. What causes this immune exhaustion? Our analysis pinpointed several key pathogenic symbionts. In the CA1 subtype, P. gingivalis and B. fragilis are significantly enriched. Functional analysis revealed that these bacteria primarily contribute to the upregulated GABA metabolic pathway that is known to mediate immune suppression and promote the proliferation of tumor cells. We also visually observed the abundant colonization of these bacteria in CA1 tumor tissues through fish experiments. This suggests that microbes may interfere with host anti-tumor immunity through metabolic products. To translate this finding into clinical application, we used machine learning algorithms to screen 24 featured genes and constructed a random forest classification model, which achieved an AUC as high as 0.938 in distinguishing the two subtypes. More importantly, we applied this model to an independent TCG colorectal cancer cohort to classify patients. The validation results showed that patients predicted to be CA1-like indeed had a significantly lower long-term survival rate than CA2-like patients. This demonstrates the robust clinical predictive value of our classification system. Comparative analysis with the findings on the core microbiome published by Wu et al., Cell, 2024, revealed that the healthy foundation microbiota reported in their study were mainly enriched in CA2 tumors, whereas CA1 tumors were dominated by pathobionts. Finally, to summarize the main contributions of this study, we proposed a new immune microbial molecular subtyping for colorectal cancer. The study reveals that specific pathogens, such as P. gingivalis, may induce immune exhaustion in the tumor microenvironment through metabolic pathways, leading to poor prognosis. The prediction model we constructed not only helps identify high-risk patients but also provides a theoretical basis for future interventions targeting specific flora to improve the efficacy of immunotherapy. We would like to thank iMetaOmics for providing a high-level platform for academic exchange. The iMeta Journal series focuses on the interdisciplinary frontier microbiology, omics, and biomedicine. We warmly welcome everyone to follow the journal and submit your work.